We are going to talk about the types of nucleic acids which are found in the cell, the RNA and the DNA. Okay, so we are going to go into the detail of these structures. The RNA is a ribonucleic acid and the DNA is a deoxyribonucleic acid. Usually the RNA is a single-stranded molecule with the number of nitrogenous bases attached. You can see the cytosine, guanine, adenine, uracil. Whereas the DNA is a double-stranded molecule where you have cytosine, guanine, adenine and thymine. So we are going to go into the detailed structure of these nucleic acids. What do you mean by these nucleic acids? They are polymers which exist as polynucleotides and made up of monomers called nucleotides. Okay, so the nucleic acids, they are polymers. Okay, and they exist as polynucleotides made up of the monomers which are called nucleotides. Now we'll go into explain this. Now they also contain carbon, hydrogen, oxygen, nitrogen and phosphorus. Another thing is that they are macromolecules. Okay, since they are polymers, they are macromolecules and we call them biopolymers. Now there are two types of nucleic acids, the DNA or deoxyribonucleic acid and RNA or ribonucleic acid. Okay, so from this slide you have to understand that they are polymers, nucleic acids are polymers, polynucleotides made up of monomer units which are nucleotides, contain carbon, hydrogen, oxygen, nitrogen and phosphorus. They are macromolecules and the two types of the nucleic acids are the DNA and the RNA. So you find that this is the structure of a DNA molecule made up of two antiparallel polynucleotide chains. So these chains are running around, they are coiled around an imaginary axis in the center, and each of these strands, they are made up of phosphate and sugar backbone. So these phosphate and sugar, they are attached together by what is called a phosphodiester bond. Now these two strands run in opposite directions, and pointing inwards from the sugars you find the nitrogenous bases. The nitrogenous bases are attached by the hydrogen bonds. Now the nitrogenous bases can be divided into purines and pyrimidines. Now the purines are the adenine and the guanine. They are larger molecules and made up of double rings. While the pyrimidines are thymine, cytosine. Uracil also is there, but uracil is found only in the RNA. Now the purines, they are larger molecules made up of double rings, while the pyrimidines, they are smaller and made up of single rings. So now you find that the always adenine pairs with thymine, guanine pairs with cytosine in this in the DNA. So the bonding, the hydrogen bonds between these nitrogenous bases keep these two strands together and makes the DNA molecule to spiral, to get a spiral appearance. Now you find that if you look at the left part of the diagram, you find that a single twist spiral of this DNA molecule. It measures about 3.4 nm and there are about 10 base pairs in this particular region in one complete spiral of this DNA molecule, and the distance between these base pairs are also 0.34 nm. You always so the distance is maintained here, and so that always you find the purine pairing with the pyrimidine and vice versa, like that. So we'll go more into the detail of this DNA molecule. we said that the DNA molecule is made up of polynucleotide strands, and each of these nucleotides, they are the monomer units. So each nucleotide unit is made up of a pentose sugar, a nitrogenous base, and a phosphate group. So you find the diagram here. The sugar here is deoxyribose sugar. Now, the nucleotide without a phosphate group is called a nucleoside. Remember, nucleotide without a phosphate group is called a nucleoside, like adenosine and guanosine. We call that adenosine and guanosine because the phosphate group is missing, and it is called a nucleoside. Now, the pentose sugars are of two types, deoxyribose and ribose. Here, the deoxyribose, one oxygen atom is less than in ribose. Okay. In the nitrogenous bases, there are two types, as I told you: purines, larger in size with two rings, and pyrimidine, smaller in size with a single ring. So in purines there are two types, adenine and guanine, and pyrimidines there are three types, thymine, uracil, and cytosine. Okay, and the bases are A, G, T, U, and C. They are represented as these letters: A, G, T, U, and C. The uracil you don't find in the DNA, you find uracil in the RNA, and you don't find the thymine in the RNA. Okay, you find thymine in DNA and uracil in RNA. So the phosphate group gives the nucleic acid the acidic nature. Now here you find the purines and the pyrimidines. The purine, the double ring structure, the adenine and the guanine, and pyrimidine, cytosine, thymine, uracil. Okay, remember that you get thymine in the DNA, you don't get thymine in the RNA, and you get uracil in the RNA, you don't get thymine in the RNA. Now in this particular strand you find DNA. How this particular picture shows how the new nucleotide can be attached to a growing strand. Now you look at the diagram on the left. So there is a formation of a phosphodiester bond between the nucleotides. By adding the 5' phosphate end of new nucleotide to the 3' OH end of the nucleic acid. So always the nucleotide is added at the 3' carbon end of the sugar molecule, and a phosphate bond is formed between the phosphate and the phosphodiester bond is formed between the phosphate and the 3' end of the sugar molecule. So you find a condensation reaction taking place, and the new nucleotide is added to the growing chain. Here is another slide which is giving you the template. So the original strand and the growing strand you find in pink, the original strand in blue, and the growing strand in the pink. So you find a new nucleotide which is added at the 3' end of the sugar molecule. Okay, and you find that there's an enzyme also involved here. The enzyme is the DNA polymerase that adds the next deoxyribonucleotide to the OH group of the at the 3' end of the growing strand. Okay, so this is the template strand, original strand, and this is the growing strand. So the strand is growing in the 5', 3' direction. Now DNA and RNA, I told you there are two types of nucleic acid depending on the type of sugar molecules involved. If the sugar molecule is in the nucleotide is deoxyribose, nucleic acid is DNA. If the pentose sugar is ribose, the nucleic acid is RNA. So DNA contains adenine, thymine, guanine, and cytosine, and RNA contains adenine, guanine, cytosine, and uracil. So this you have to remember. Okay, if DNA contains deoxyribose sugar, RNA contains ribose sugar, and the nitrogenous bases are in the DNA and the one found in the RNA. Here you find a diagram of the deoxyribose sugar which you find in the DNA, one oxygen atom less, and here you have the ribose sugar. Now this is a Watson and Crick model of the DNA molecule. So as I told you, it has two anti-parallel polynucleotide chains spiral around an imaginary axis forming a double helix. Okay, two anti-parallel polynucleotide strands that spiral around an imaginary axis forming a double helix. So two sugar phosphate backbones, they run in opposite direction, and this arrangement is referred to as anti-parallel. So sugar phosphate backbone on the outside of the helix and the nitrogen bases are paired in the interior of the helix. So the two strands are held by hydrogen bonds between the paired nitrogen bases. So the two strands are said to be complementary to each other. Okay. So these pairs are known as complementary base pairs. In this original double helical structure, one complete turn consists of 10 base pairs. We have discussed all that. So the two strands, remember that it's a double helix. Okay. The DNA molecule is a double helix made up of two anti-parallel polynucleotide chains, and these chains, they run in opposite direction, they are anti-parallel and they are complementary because these nitrogen bases are paired in the interior of the helix and bound by hydrogen bonds. So these two strands are said to be complementary. The same thing is given here in this diagram. Now functions of DNA. DNA stores and transmits genetic information from one generation to the next generation, and next one, it is stores genetic information for protein synthesis. So DNA is very important. It is found in the nucleus of the cell, and it is involved in storing and transmitting the genetic information from one generation to the next, and it has genetic information stored for the protein synthesis. Now we come to the RNA. Usually RNAs are single-stranded molecules, okay, and composed of ribonucleotides, and they contain the bases are uracil, cytosine, guanine and adenine. Complementary, usually they are single strands, but they can form complementary base pairing between two RNA molecules or within the same molecule may occur. Okay, so complementary base pairing facilitates a three-dimensional shape essential for their functioning. So adenine binds with uracil with 2 hydrogen bonds, and guanine binds with cytosine with 3 hydrogen bonds. So there are 3 types of RNA: messenger RNA, tRNA and rRNA. Okay. Now mRNA is a linear molecule, least abundant type of RNA in the cell. Okay, it's a linear molecule, least abundant. Now functions: copies the genetic information in the DNA molecule as a sequence of nitrogen bases. So it copies the information from the DNA. Okay, the information from the DNA is actually transcribed onto the RNA, and it brings the information out into the cytoplasm where for protein synthesis to take place. It transports the genetic information from the nucleoplasm to the site of protein synthesis. Okay, from the nucleus through the nucleopores. Okay, so it is a linear molecule, least abundant. It carries the information from the nucleus into the cytoplasm where protein synthesis takes place. Now transfer RNA, it is the smallest RNA molecule, linear, but forms a 3-loop structure. Now it's a clover shaped structure where it has 3 loops, and you find one, and here also you have 5 feet and 3 feet, and it's a single strand which is folded on itself to form a clover shaped structure. So this is the amino acid binding site. Okay, so where the amino acid, you find the tRNA molecule, this is the it carries the amino acid at one end, and at the other end you find the anticodon. So it brings the amino acid and matches the anticodon with the mRNA. Okay, so that the amino acids, the protein synthesis can take place. Now rRNA, the most abundant type of RNA. Now this is RNA is found in the rRNA is found in the ribosomes. Ribosomes are made in the nucleolus, in the nucleus. So rRNA is a complex irregular structure. You don't have a particular structure. It provides a site where polypeptide chains are assembled. So now you find that this ribosomal RNA. Now, so all these three types of RNA, they are involved in protein synthesis. So the mRNA brings the code from the nucleus out. Can you see this strand, mRNA strand coming out of the nucleus with the codon on it, and then the tRNA, it binds to the amino acid, picks up that particular amino acid, and with the anticodon, you find the U anticodon in this particular one. Then here you find the already two tRNA are here at the site of proteins, and they are matched. The anticodons are matching with the codons on the mRNA. So you find AUG, so protein synthesis has been started here, and they bring the amino acids, and the amino polypeptide chain is also formed. So all these three RNAs, they are involved in the protein synthesis. Difference between DNA and RNA. RNA is a double-stranded molecule. RNA is a single-stranded molecule. DNA consists of A, T, G, C and absence of uracil, while RNA consists of A, U, G and C and absence of thymine. So sugar molecule in RNA is ribose. In DNA it is deoxyribose.